Hi there, welcome to this video. So this is about D3, which is all about the sources of error. So a source of error means where could there have been an error in the way you did your method, which could have caused some anomalous data. So anomalous data is basically like data that isn't right, that isn't accurate. So it says this should be done qualitatively only, so you don't have to do any calculating in this part. And the students who have no anomalous data should still have explain to clarify this in their written evidence. Okay, so even if you don't have any anomalous data, even if you think your data is all really good, you still have to do this section. So the heading for this section is evaluation. And first of all, we need to be looking at each experiment we did. So don't just think of one source of error of one experiment. We need to think of each experiment that we did. So before we start, let's just have a think about what experiments we did do. So obviously you're only going to do this for the experiments that you did, which is different for each person. Lots of people started off with the little dipsticks in the urine. So they were called reagent test strips. So just in our planning, I'm going to do it as a spider diagram, but obviously you'd write it up probably as paragraphs. Okay, so we did reagent test strips. Lots of you then did Benedict's. And the first Benedict's that we did was called qualitative Benedict's. The next one lots of you did was, can we insert a new page break. The next one lots of you did was the Biuret, which was testing for protein. The next one we did was something, all the microbiology. So the microbiology, we could put that into one. So we obviously made our streak plates and we plated onto two types of agar. So we call that selective plating. And then we looked at the colony morphology. And then we did the quantitative Benedict's. Now, a few of you also did ion tests, but not many of you, so I'm not going to cover that in this video. Okay, so let's go back to what we needed to include. So it says consider each of the experiments you did. So we've kind of got our headings and write a paragraph for each one, including, do you think you had any anomalous results or not? So that's up to you. If you did have anomalous data, what was it? What could the sources of error be? How might they have affected your data? And what should you do in the future to reduce these sources of error? So let's have a think about our first one. So the first one we did was the reagent test strip. So let's have a little think about what the sources of error could have been. So what you do in a reagent test strip is you've got your sample in the tube and you stick into it the little reagent test strip with the little squares on it. And you're meant to dip it in and then wait 60 seconds. So the first source of error is if you don't dip this all the way in. So say you only dipped it in that far, you haven't actually dipped these top squares. And I did see that as I walked around the room sometimes. And therefore you won't get any colour changes on these top ones. And you won't even know whether you didn't get a colour change because it was a negative test and it was meant to be white, or whether you just didn't dip it and therefore it didn't have any substance to make it change. So a source of error could be, for example, that you didn't dip the whole strip into the liquid, okay? And therefore the top test pads don't change colour. and therefore you would get an inaccurate result. Yeah, you get the wrong result because you would think that that square is white. So you think that it's white, perhaps it should be a different colour. Okay, so if we go back to the PowerPoint, it says, what was your anomalous data? So your anomalous data could be something like, I did a reagent test strip looking for glucose. However, it said it was negative. However, when I tested with tested for glucose using other methods such as qualitative and quantitative Benedicts, it said it did have glucose. So I think my glucose test using the test strip was wrong, for example. Yeah. So what was your anomalous data? What could the source of error be? So it could be that you didn't dip your test strip all the way in. How would this affect your data? Well, the little test pads haven't absorbed any liquid, so they didn't change colour as they should do. What should you do in the future to reduce this source of error? So you could then talk about how in the future I would make sure I dip it all the way in. So all test pads are submerged. Okay. Now the next thing that you're meant to do with these reagent test strips is you're meant to read the colour after 60 seconds. Okay, so you take the test strip out and you lay it flat, and you should read it after 60 seconds. So if you're looking up the colour of this pad, and then the colour of this pad, and then the colour of this pad, and then the colour of this pad, even if you used a stopwatch to try and time 60 seconds, by the time you're reading the pad at the bottom here, more than 60 seconds have passed, haven't they? So not reading the colours after exactly 60 seconds. Now the impact of that is that they may have changed colour again. And therefore you're going to get an inaccurate result. If you did it again, so if you were to do it again, what could you do differently? Well, what you could do is perhaps take a photo at 60 seconds so that when you read the colours off, it was the colour that it was at 60 seconds. Yeah. Another thing that you're meant to do is you're meant to lay it flat when you read off it. So if you didn't lay it flat, then what could sometimes happen is that the substance from one pad moves to another or diffuses to another pad, and then it affects the colour of that pad. So you get, obviously, your inaccurate result again. What could we do about that? Well, we lay it flat. So if we did it again, I would lay it flat. Yeah, so hopefully this starts to give you an idea of what this section is about. Okay, the next test lots of you did was the qualitative Benedicts. So for qualitative Benedicts, what you did is, first of all, you had your blue Benedict's solution. So you had your bottle with your blue solution in, which was your Benedict's. And you put that with your sample. So you put your sample in here and you put your Benedict's in here. And the first thing that you did is that you had to measure the sample and the Benedict's. So the first thing that could be a source of error is inaccurate measuring of the liquid volumes. So you might have inaccurately measured the Benedict's or the sample. Now you used Pasteur pipettes. Now, you have now worked out your percentage uncertainties. So what you could do is you could bring in those results to this discussion, and you could say that the percentage uncertainty of Pasteur pipettes is higher than graduated pipettes because the resolution isn't as good, because the interval between the markings are 0.5 millilitres, whereas on a graduated pipette it's 0.1 millilitres. So if you've got inaccurate measuring of these liquid that you're putting in here, it could mean that the colour change was inaccurate. Now, when we do qualitative Benedicts, we get like this scale of colour that it goes. So the first colour is blue, and then we've got green, and then we've got yellow, then we've got orange, and then we've got red. So if it goes red, that means that there's a high concentration of glucose. If it goes blue, there's no glucose, it just stays the colour that it was in the bottle. If it's green, there's a low concentration of glucose, and then the other colours are like a scale. So a little bit higher concentration, a little bit higher concentration, high concentration. Okay. Sometimes when you look this up, it says of reducing sugar. Glucose is a reducing sugar. Okay. So if the colour change was inaccurate here, if you would then think that there's a different concentration of glucose to what there actually is. So you would make the wrong conclusion about the glucose concentration. And if you did it again another time, what could we do to prevent that? Perhaps we should have used a graduated pipette next time, because it has a higher resolution, because it has a lower percentage uncertainty. Yeah, so it's got a better resolution and a lower percentage, oops, draw a box on that, uncertainty. Rubs out off the side. Okay. So that's one thing you could have done wrong. Another thing that you could have done wrong, another source of error, could be that maybe when you put the Benedicts and the sample together, you didn't mix them. Yeah, so if you didn't mix the Benedict solution and the sample together, then they won't have reacted fully. And therefore, just like the other one, you might have got the wrong colour, and then you may have made the conclusion about the wrong glucose concentration. So if you did it again, how would you avoid that? Is to make sure that you definitely do mix. Usually to mix, you would put a bung in the top of the test tube and then invert the test tube. So it goes like upside down, right way up, upside down, right way up, upside down, right way up a few times to make sure it's fully mixed. So you could say 5 times. Okay, to make sure it's fully mixed. Once you'd put your Benedicts and your sample in together, the next step that you did is that you stood it in a water bath. And the water in this water bath needed to be really hot. So it might be that you didn't heat. Another source of error is that you didn't heat it enough. Now, the way we made our water bath is that we just boiled the kettle and poured it in. So it could be that you boiled the kettle 5 minutes before and the kettle had already cooled, so the water was not hot enough. Yeah. It could be that the water in the beaker cooled down. So by the time we got to the end of the 5 minutes, it wasn't the temperature it was at the start. Yeah. So if that was the case, then if it's not hot enough, then the Benedicts won't react. And the result of it not reacting, again, goes to the same as here, wrong colour, wrong glucose concentration. Okay, if we did it again, how could we do the heating part better? We could use an actual water bath, not a beaker filled with hot water, but an actual water bath that has a thermostat in it. And a thermostat makes sure that the temperature stays what you set it at. So you could say thermostat maintained at 95 degrees C, and that would make sure that it's definitely staying at the right temperature, so it definitely works. The next one that we looked at was, not that one, the Biuret reagent. Where has it gone? Here we go. So Biuret is very similar in method to the Benedict's, but you don't need to heat it. So the Biuret solution, what you did here is you added some Biuret in there and you added your sample in there and then you mixed it and then it changes colour. And this time the colour that we are looking for was purple. So if it turns purple, it contains protein. If it doesn't go purple, it doesn't contain protein. So what could have gone here? So the same as the qualitative Benedict's, in that you could have measured inaccurately. So you can use what we discussed in that one. You didn't need to heat it, so the heating part is not relevant, but you did need to mix it. So same as qualitative Benedict's, you might not have mixed it correctly. Now the Biuret had another problem. Lots of people had an unclear colour, okay? And this is because the urine sample was cloudy, and it doesn't just contain like water and protein, it actually contained other molecules as well that interfered with the test. Okay, so it might be the case here that there would be another experiment that's better, because Biuret actually didn't work very well with our urine sample because there was obviously something in the urine that was interfering with the colour and it didn't make a very clear purple. So we didn't get a very clear result. So in the future, perhaps if we did it again, perhaps we need to use a different test. So, for example, we did use the reagent test strip to try and back up our findings, but you can research other things that you could have used as well, other tests. The next one that we did was all the microbiology. So what are the sources of error in the microbiology? Now, there's quite a lot of things that went wrong in this. The results were not very clear. So first of all, it could be in your streak method, because if it's not done correctly, then you get no clear individual colonies. So then links to this one, it was then very hard to identify the colony morphology correctly. By not doing it correctly, what I mean by the streak plate method is that you should have done a streak there, which would have loads of microbes in, then one there that would have less, then one there that would have less, and then one into the middle. Now, if that middle streak overlaps these others, it picks up too many. So after it's incubated, you should get loads and loads of colonies in the first streak, then less in the second, less in the third, and just a few in the middle. And those ones in the middle should be really separate and individual. So they should be really easy to identify, like the shape, the form, the elevation, etc. Okay, but if we didn't do that streak plate correctly, then we've got too many bacteria in this middle section and the colonies all merge together and you can't look at the shape of them very clearly. So maybe the streak plating went wrong. Now, if you did it again to improve that, basically you need more practice. Okay, it's hard to see where you're putting the streaks, but you get the hang of it the more you do it. So it's just really a case of practicing. Another source of error was that when we looked at our plates at the end, most of the plates, you have to talk about your individual plate, but most of the plates were contaminated. When we looked at the plates at the end, they had multiple species growing, okay, all in different colours, different colony morphologies. And that meant that you didn't know which was in the original sample and didn't know which one that you should have been identifying. Didn't know which to identify. So somewhere along the way, it got contaminated. Okay, so the problem was contamination. Now, the reason when it might have got contaminated is that you might not have had good aseptic technique. So you could talk about the aseptic technique that you used, such as working next to the Bunsen burner, flaming the neck of the bottle, sterilizing your loop, etc., etc. So it might have been that your aseptic technique wasn't good, or it might have been to do with maintaining the integrity of our samples. Because our samples should have been kept sterile, and maybe they got contaminated because you were using those same samples for all your experiments. So you didn't sterilize the pipette for your Benedict's test when you took some of the sample out. So maybe you contaminated the sample, although most of you didn't do the other tests on sample 3 because this was of sample 3. And most of the other experiments you just did on sample 1 and 2. So you might not have contaminated it, but you might have done, somebody might have done. Also, we were sharing samples with other people in the class, so somebody else might have contaminated it. Yeah, so maybe we didn't maintain the integrity of our samples well enough and they got contaminated at another time, or maybe our aseptic technique wasn't good enough and we contaminated it while we were making our plates. We don't know. Okay. With the selective plating, I think the source of error with that one was the same. It was to do with the contamination. Okay, so what you should have been able to see really clearly is whether your species grew only on the nutrient agar, only on the malt extract agar, or whether it grew on both. But because you didn't know which species you were meant to be looking at on your plate, because lots of species grew, it was hard to work that out. It wasn't that the selective plating went wrong, it was just that you didn't know which colonies to look at. So if we did it again, to overcome this contamination, we need to keep our samples sterile. Okay, we need to only use them once, and we need to have really good aseptic technique. The next one that we all did was the quantitative Benedict. So this quantitative Benedict's was slightly different because what we did is we had some known concentrations of glucose, if you remember, and we put them in the tubes 0, 10, 20, 30, 40, 50. And we added our sample to each of those, so our urine sample. And in our urine sample, we didn't know unknown glucose concentration. Oh, we put that in a separate tube, sorry. Let's get that arrow there. Oh, we want to get rid of the whole lot. Oh, what have I done? Let me click off that. Where's it gone? There we go. So in there, we didn't put our urine sample. That's what I'm trying to say. Doesn't want me to rub it out. Never mind. I just cross out. So we didn't put our urine sample in there. I'll talk about that in a minute. We put in the bottom of here our known glucose concentrations, and these are the glucose concentrations in millimoles. okay? And then in the top, we added our Benedicts, and the Benedicts was a different type of Benedicts to what we used before. This was called quantitative Benedicts. So the first thing that we needed to do in this experiment is we need to measure using a pipette the known concentrations of glucose and the quantitative Benedicts. So the first source of error could have been in measuring those volumes incorrectly. And for the details of that, you could go to the Benedicts, the qualitative Benedicts that we talked about. But remember, this time we measured our known concentrations using a graduated pipette. Yeah, so that actually already has a high resolution and a low percentage uncertainty. So that should already be quite accurate. Okay, but then we added the Benedicts using a Pasteur pipette. So that might have had more error, okay, because that has a lower resolution or a higher percentage uncertainty. So it's not such a good thing to use for measuring. Okay, so we might have had a source of error in measuring these volumes inaccurately. Okay, that might have been a source of error. So if we did that again, we need to be extra careful. And if we did it again, perhaps we should measure our Benedicts with the graduated pipette, and that would be better because it has a higher resolution. Now, the next thing we did, if you remember, is we transferred these into the little cuvettes, and we put those cuvettes one at a time into the colorimeter. Remember the little colorimeter machine, and you put it in here, and then it had a little screen and it gave you the number. Yeah? So we added our samples into a colorimeter. Okay, actually there was a step in between, I should have said. We heated them. So we could talk of a source of error of the problem with the water bath, and that will be the same as we discussed in the other Benedicts. So we used a beaker for our water bath. That's not the best way of using a water bath. We can't guarantee the temperature. If it's not hot enough, the Benedicts doesn't work. It doesn't change to the right colour, and therefore we're not going to get the right colour, so we're going to get the wrong colour. They all go blue in this one. We get the wrong colour blue, so we're actually going to end up with the wrong calibration curve. Okay, so we put those into the colorimeter. Now, when we put them into the colorimeter, what blank we use made a difference. So another source of error could be to do with what we used as a blank. Now, you might remember that at first, we tried using the 0 millimole as the blank, but the results didn't follow the right pattern. So then we start using water as our blank, and then we did start getting the right pattern. Okay, so the blank made a difference. We made a mistake here, and then we corrected it by changing it to water as the blank, because then when we put all the other concentrations in, we started to get the right colour. Okay, so then you might remember what we did. We go up to here, is we used those known concentrations to plot our calibration curve, and our calibration curve looked something like this. Okay, and then what we did is we measured our unknown, our unknown urine samples where we didn't know what the concentration was. So we had transmission up the side of our graph and concentration of glucose on the bottom, and if we put our urine sample in the colorimeter and it gave a reading of 20, we went to 20 here, we drew a line across till it hit our curve, down until it hit the concentration, and we read the reading of the concentration here. And that gave us what we think was the concentration of urine sample 1 and urine sample 2. So a source of error in doing this process could be that we read the concentration of urine samples incorrectly. So did you draw these lines correctly and accurately? Now, what you might have found is that that was like marked with little markings on there, but sometimes you had to predict numbers that were in between, and you weren't able to do that accurately because it didn't have all the little markings in between those. So that would be a source of error because you couldn't read off the graph accurately. So if we were to do it again, how would we read these off here and off this scale as well, off this axis as well? How would we read these more accurately? So if we did it again, what it would be useful to do is to draw more subdivisions on the graph axes. And then what you'd be able to do is you'd be able to read the numbers off the graphs much more accurately, and it wouldn't be such a problem. Okay. Let's just think, let me think this through if there's any other reasons why it might have gone wrong. I think for most of you, oh, I know, I've just had another thought. Right, so when you heated these test tubes up and then you left them to settle, you put them in a test tube rack, and you might remember that the way this type of Benedict's works is that it produced a precipitate, like lots of little dots floating in the liquid, and what you had to do is you had to wait for that precipitate to settle. So what happened after a while is that that precipitate settled to the bottom and you were left with like a clear liquid at the top. And then you wanted to pipette that clear liquid using your pipette. That doesn't look like a pipette, but you want to take that clear liquid and put it into the cuvette. Okay, so you were using a pipette to transfer that liquid into the cuvette, which is the little plastic square pot that you put into the colorimeter. Yeah. Now, some of you, I think, accidentally used your pipette and you put it right down to the bottom. And so you sucked up some of this precipitate and it made the mixture more cloudy and it gave you inaccurate readings. Okay. Some of you didn't wait long enough for the precipitate to settle, so there was still precipitate in the liquid. So even if you took it from the top here, there were still some bits floating around and you picked them up. So how do we write that? So what we could say is a source of error could be that you didn't wait long enough for the precipitate to settle, or perhaps you put the pipette too deep when you took the sample, for the cuvette. And either way, what that meant was that you had precipitate in the cuvette. And the effect then of that is that it was too cloudy. And therefore, when you tried to measure the transmission, what happened is not as much light went through, so you got a low transmission reading. And therefore, your graph would have been inaccurate. And therefore, when you read, I've run out of space, sorry, I'm going to move over here. Use this little bit of space over here. So you got a lower transmission reading than you should have. So that meant that your calibration curve was inaccurate. So then when you read your urine samples off that curve, They were inaccurate. Now, if we did it again, how would we avoid that? So if we did this again, we need to make sure we don't pick any precipitate up. Okay, really, really important. Make sure we don't pick up any precipitate. All right, so hopefully that's helpful. Let's go back to this and just make sure we understand how we're writing it. So we're writing a paragraph for each experiment that we did. Do we think we had any anomalous results? In which case, what could have gone wrong? And you might just say, I'm not sure if it was an anomaly, but I'm not confident that it was totally accurate. So the sources of error could have been, and then go back to what we've discussed, they would have affected my data by what we've discussed. If I did it in the future to reduce the source of error, I would do it like this. Okay, even if you think you didn't have any anomalous data, it doesn't mean your data was perfect. So I would say something along the lines of, even though I don't have any obvious anomalies, I don't know if my results are completely accurate, and therefore I would still discuss the sources of error that we discussed. But you could say, you know, for example, I don't think that I did measure it inaccurately, but I could make it even more accurate next time by using this pipette. I don't think that my water bath did cool down, but to make it even better next time, I could use a proper thermostatically controlled water bath, etc. Okay, so there's still improvements to make, even if you don't think you have anomalies in your results. And that's it.